to share with you some of the recent results that we've obtained visualizing whole tissue cultured cells in the frozen hydrated state, and to describe in a very general sense the method of plunge freezing for cryofixation that I use to both image cells that have been free substituted for routine electron microscopy and also to image frozen cells in a cold stage. I hope that you come away from this talk with a general knowledge of how to cryofix cells for free substitution or for cryoimaging, have a general sense of the advantage of these techniques over routine chemical fixation, and also look towards the promise of intermediate and high voltage electron microscopes for imaging thick frozen cells. If I could have the first slide, please. First of all, why cryofixation? Well, one can think of at least three criteria which must be satisfied for the optimum fixation of biological tissue. First, the fixation regimen should act quickly and immobilize all molecules and atoms in the cell. Third, a fixative should not alter a cell's morphology that was present before fixation. If I could have the next slide. This slide illustrates how routine chemical fixatives often fail to meet these three criteria. Chemical fixatives often alter the cell's morphology, as seen by the wavy microtubules in this mitotic cell. Microtubules should be straight, and the presence of myelin figures within the cell. These artifacts may be due to the fact that chemical fixatives act by diffusion, so fixation occurs at different rates within the cell. No chemical fix alone can fix all molecules in the cell, and thus different combinations of chemical fixes, for example, glutaraldehyde in association with osmium, must be used, although many enzymes maintain reactivity even after glutaraldehyde fixation. Cryofixation, or the rapid freezing of a sample, has the advantage in that it rapidly fixes almost instantaneously all molecules and atoms in the cell without alteration of cell morphology, thus avoiding some of the artifacts shown in this slide. Next slide, please. I use the plunge freezing method to cryofix monolayers of cultured cells for either free substitution analysis or for directly imaging frozen cells in the cryomicroscope. I use PTK cells, that's the cell type I'll be talking about today, which is a mammalian epithelial cell line. They're grown onto carbon-coated, form-bar-coated, carbon-stabilized gold grids. Next slide. Immediately before freezing, the grid is removed from the culture medium and blotted in a humidity chamber to remove the majority of the fluid on the grid. This blotting procedure was critical to obtain a thin enough sample to ensure both optimum cryofixation and imaging in the cold stage. After blotting, the grid is then immediately transferred to a freezing apparatus, shown on the next slide. This is an overview of the freezing apparatus that I use. It's a plunge freezer that is modeled after a design by Dr. John Murray at the University of Pennsylvania. It consists of a vertical slide rail that guides the fall of a mounted pair of forceps that clamp the specimen. The forceps are surrounded by a plexiglass humidity chamber in which cold, humid air is continually pumped in to prevent drying of the sample before freezing. Next slide. The forceps are mounted over a vessel of ethane, which is liquefied in a liquid nitrogen cooling bath, and I add fresh ethane before each freezing run. Next slide. The blotted grid is then quickly plunged into the ethane using a quick release mechanism, and the ethane is held directly above its freezing point. Next slide. The frozen grid is then quickly placed in a transfer vessel containing liquid nitrogen, where it remains at this temperature until either viewing directly in the cryo stage or after further processing using free substitution. The plunge freezing method has limitations in that relatively thin samples can be cryofixed optimally. These are samples less than about 5 microns thick. So that to assess the quality of freezing using the PTK cells, I then processed the grids further using free substitution and either embedded them for routine electron microscopy or dried them from carbon dioxide using the critical point method. Next slide. This slide illustrates the free substitution apparatus we use, which can be assembled in the lab with a modest investment. It consists of a dewar filled with liquid nitrogen to cool a specimen chamber containing the frozen samples. Next slide. The specimen chamber is held at minus 90 degrees using an adjustable temperature controller and rheostat. I free substitute my samples in 1% osmium tetroxide and acetone for 3 days at minus 90 degrees, after which time the samples are gradually warmed to room temperature and either infiltrated with resin for routine section microscopy or dried from CO2 using the critical point method. Next slide. Kent McDonald and Mary Morphew of the Boulder High Voltage Lab have looked extensively at different free substitution protocols, and this slide illustrates some of their results. They high pressure froze Drosophila embryos and free substituted them in a variety of cocktails, including 1% osmium and acetone, 2% glutaraldehyde and acetone, 1% osmium and methanol or methanol alone. They found that 1% osmium in acetone gave the best free substitution and resulted in tissue that had nice, dense, and uniform cytoplasm, a good definition and contrast of plasma membranes, as can be seen by the plasma membranes between these two adjacent cells, as well as the preservation of microtubules and organelles within the cell. However, the tissue that were free substituted in methanol or glutaraldehyde and acetone had a general extraction of the cytoplasm and no clear definition of membranes. Here are two adjacent cells, and you can't really resolve the membranes between the two cells. The bottom two panels show tissue that were free substituted in 0.01% osmium in acetone and embedded in either epon araldite or LR white. And these investigators have had success immunolabeling sections embedded in LR white, and even with this low concentration of osmium, the tissue is well contrasted and preserved. Thus, fast freezing methods followed by free substitution not only give us the advantage of optimum fixation, but may also preserve sensitive antigenic sites within the cell. Next slide, please. However, fast freezing and free substitution may also introduce artifacts that are not present in chemical fixes, and an example of this is shown on this slide. This is a section through a plunge-frozen, free substituted PTK cell, which has been improperly frozen. And one can see the overall effect is a reticular network that's present in the nucleus and in the cytoplasm, which is characteristic of gross ice crystal damage. And I found that the blotting of the grid before freezing was critical to obtain a thin enough sample to ensure optimum cryofixation. Can I have the next slide, please? This is a section through a plunge-frozen, free substituted cell, which has been blotted and thus properly cryofixed. This is at a comparable magnification of the previous slide, and one can see that there is good preservation of organelles, such as rough endoplasmic reticulum and mitochondria, and the cytoplasm is dense and uniform. Next slide. At higher magnification, there is Little evidence of ice crystal damage, which would be visible as a reticular pattern in the cytoplasm. Here it's very dense and uniform, as well as clear halos around organelles if there was ice crystal damage. Next slide. Cells that have been plunge frozen and free substituted and critical point dried similarly have good fixation, as shown in this whole mount, low magnification of a plunge frozen free substituted cell. And these cells are well fixed even in the thicker parts of the cell, including the nucleus and perinuclear region. Next slide. At higher magnification, organelles such as mitochondria and vesicles are similarly well preserved and embedded in a dense cytoplasmic matrix. Because of the results using free substituted material, we were convinced our cryofixation method was a good one and felt confident to image whole frozen cells in the cryo stage. Next slide. This slide illustrates the cold stage that was used in the studies that I'll describe. This cold stage was designed and built by Mr. George Ray, who recently retired from our lab. This is a top entry stage, which has a point-to-point resolution of greater than 8 angstrom. The specimen, which sits in the center of the stage, is cooled by nitrogen gas, and we routinely observed our frozen samples at minus 150 degrees centigrade. Next slide. An advantage of the higher voltage electron microscopes, as was mentioned earlier, is that one can image thicker samples with improved resolution and penetration of the electron beam. Conventional microscopes would require an increased electron dose to penetrate a sample this thick, and thus a frozen sample would damage very rapidly. This is a micrograph of a thin section, or a thin portion of a PTK cell embedded in relatively thick ice. One can see the outer edge of the cell, as well as hints of structure in the cytoplasm, stress fibers, and shadows of organelles within the cell. However, the details within the ice are not evident for several reasons. Imaging a frozen sample is very different from imaging a dried cell or a stained section. Because the cells are embedded in ice and not stained, they must be imaged at phase contrast, and very low electron doses are required to prevent damaging the specimen. Next slide. This slide illustrates the importance of phase contrast focusing using frozen hydrated samples. Panel A shows a thin portion of an elongating PTK cell with the image taken at focus, and panel B shows the same area taken at 20 microns under focus to obtain phase contrast. And at phase contrast, one can see the details of the organelles and resolve individual microfilaments. Next slide. An online image processing program and video system that we have on our microscope greatly facilitates imaging our frozen samples at very low electron doses and is very helpful in determining the amount of underfocus to obtain phase contrast. Our actual image capture is done using a fast film, a Kodak SO163 film, using 2-second exposures. Next slide. This slide illustrates the importance of low-dose imaging and shows the dramatic effect of the electron beam on frozen samples. This is a low magnification of a frozen hydrated cell, which has been exposed in this area at a very high electron dose of over 1,000 electrons per square angstrom. We consider low doses to be 1 to 2 electrons per square angstrom per second. On the left side, where the cell was spared such a high dose, one can see organelles, mitochondria, and a delicate cytoplasmic matrix. However, in the area of higher radiation, one can see blisters and bubbles and these larger structures. I have watched this damage happen before my eyes, and it starts out with these small blisters and bubbles, which then coalesce to these larger structures and holes in the cell. Next slide. At higher magnification, this beam damage is even more apparent. Panel A shows a thick portion of a frozen PTK cell after exposure to a total electron dose of about 5 electrons per angstrom squared. After a total dose of over 100 electrons per angstrom squared, one can see this characteristic bubbling of the sample and a complete loss of fine structure. This type of beam damage is unique and characteristic of frozen hydrated material. Next slide. In contrast, PTK cells which have been dehydrated in critical point dried do not damage by bubbling when exposed to the same electron dose of over 100 electrons per square angstrom. Next slide. Another important aspect of cryoimaging frozen samples is documenting the state of the ice using electron diffraction in the affected area. The top two panels show a thick organelle-rich region of a frozen hydrated cell and its corresponding electron diffraction pattern. This is taken at a cold stage temperature of minus 150 degrees. The diffraction pattern, although it might be difficult to see, is characterized by 3 rings around a bright central maximum. If the cold stage is allowed to warm, there's a change in the electron diffraction pattern to one that is characteristic of crystalline ice. Next slide. In contrast, PTK cells which have been critical point dried have an electron diffraction pattern that differs from the frozen hydrated material and is characterized by 2 diffuse rings around a central maximum, which do not undergo transitions to a crystalline form upon stage warming. Next slide. One can then measure the ring diameters from the electron diffraction patterns to calculate the d-spacings that are present in the images. And the d-spacings are measures of the actual distances, molecular distances between lattice planes in the material, and can give us useful information on the structural nature of the sample, whether it's crystalline in nature or whether the atoms are arranged in a purely random fashion. We analyzed frozen grids of pure water and measured the d-spacings and found that similar d-spacings were present in the thin frozen cells in which detailed ultrastructure was visible. In contrast, dried cells or carbon films, which contain only 2 diffuse rings, have d-spacings which are different from the frozen hydrated material. We concluded from these data that ice was present in the frozen cells and that these were different from the either dried cells or pure carbon films. Next slide, please. Okay. When taking all of these technical considerations for cryomicroscopy into account, we've been able to obtain images rich in cytoplasmic detail, as seen in this peripheral region of a thin frozen PTK cell. Next slide. In thicker organelle-rich regions of the cell, mitochondria and filaments are well preserved and embedded in a dense cytoplasmic matrix. Next slide. In thicker parts of the cell, the cytoplasm is very dense yet highly structured. We are convinced that these cells are not dried for the following reasons. Next slide. If a grid is allowed to freeze dry in the microscope at minus 90 degrees for 15 hours, it becomes covered with a protein and salt precipitate. And this is due to the fact that the grids are plunged directly into the ethane without prior rinsing. And what's left behind are salt and protein precipitate from the culture medium. And the electron diffraction pattern characterizes the crystalline nature of these salts. Next slide. The cells are also not air dried. Occasionally, Air-dried cells can be found on the edges of the blotted frozen grids. These air-dried cells are easily recognized and have a unique morphology, which is characterized by a complete lack of fine structure within the cell. Next slide. And finally, frozen hydrated cells have a morphology which is very different from critical point dried cells. Higher magnifications of this area and this area on the next slide show a dramatic difference in the structure of the cytoplasm. Panel A shows a high magnification of the cytoplasm from a frozen hydrated cell, and panel B shows an equivalent area from a cell which has been plunge frozen, free-substituted, and critical point dried. Next, I would just like to conclude by saying that although this monolayer plunge freezing technique is limited to relatively thin samples, it can be applied to many different cell systems to answer many important biological questions. Even if you don't have access to a cryostage, the plunge freezing method followed by free substitution can give elegant fixation of samples, either for routine electron microscopy and immunoelectron microscopy. However, cryoimaging of these frozen samples give us an opportunity to visualize cells which have never been exposed to chemical fixatives, dehydration, or stain, and allow one to visualize a sample in a manner which is close to its native state. We are encouraged by the clarity and wealth of detail in the cells and feel that there's a great promise for the higher voltage electron microscopes, the intermediate and high voltage EMs, to visualize thick frozen cells. Thank you.
Gaining some time, I'd like to open up for questions not only for Eileen but also for Lee. You want to come up too if there's some questions for you? Let's take a few minutes for questions and then we'll have a coffee break. Carolyn.
One comment and one question. The comment, our laboratory is starting an effort to get a high-pressure freezer. If there's anyone in the room with a serious interest, please talk to me today. And the question, Eileen, have you ever tried any cryoprotection with frozen hydrated material?
No, they're frozen directly from the culture medium, which may have a cryoprotective effect in that it's very high in concentration of proteins. So maybe proteins in the medium will help fixing. But I block them so that they're very thin samples to begin with.
I had a question for Lee, actually. I was wondering whether it would be possible. You showed specimens where you were outlining sort of surface using like nanobodies and little particles. Could you actually do something almost equivalent of negative stain on the surface, something that would have either at random but be much closer together and get an even more accurate sense of the contour? Is there a methodology that would allow you to sort of really, in other words, you're doing particles at a certain station, but in principle, the closer they got, it would increase your problems of construction if you would get higher resolution. Is there a theoretical limit as to how you could do that? No. Is there a strategy you could use to do that?
In order to do this using the stereo method, you have to have something that's particular. You have to be, your eye has to be able to identify, the computer has to be able to identify the same particle in the picture. If it was uniformly covered with a negative stain on a uniform sheet, then it wouldn't work for it.
Be able to recognize very close details. I'm sorry, I may not have stated that very well. Well, you have a stereo pair, and you have to solve the problem, what the correspondence problem. You have to know that this particle in this image is the same as that particle in that image, and then your brain will properly position that in the Z direction, or the computer will properly position it. But if it's a smooth sheet, which is the limit of making the particles closer and closer together, then you can't solve that correspondence problem except for the very gross contours of the sheet. So you can certainly bring them closer and closer together, but there's a limit when then you begin to lose it. We now use much smaller gold particles. That particular set of slides, which I tend to use in this kind of a lecture, was 15 nanometer gold particles, and we use 5 nanometer gold particles, and you can get them more closely spaced. Of course, if you're staining a particular antibody, the distribution of that antibody is what's going to determine the distribution of the gold particles. Yes.
You know what the cooling rates were for your plunge frozen cell?
No,
Maybe you want to repeat the question.
the question was whether I know the cooling rates, and I haven't measured the cooling rates for them.
Why did you use ethane?
I use ethane instead of propane or another, because the ethane will sublime. If you have a frozen layer of ethane on your samples, it will sublime in the microscope. And so if you're introducing, after you freeze your sample and put it into liquid nitrogen before transfer into the cryo stage, any frozen ethane will sublime away.
And the other question is, how do you distinguish, how do you find out if your frozen hydrogen samples are not, don't have ice damage?
Well, I monitor,
Ice crystal of maybe 10 angstrom or 20 angstrom.
the question was, how do I determine whether the frozen samples have ice crystal damage? The ice crystal damage, as far as the gross ice crystal damage, the large reticulate, is obvious even in a frozen hydrated sample. But the smaller crystals that you're talking about, I haven't been able to measure that. I monitor the state of the ice by electron diffraction, so I can determine if there's crystalline ice that's present in the cell. And I found that in the thicker regions of the cell, I've never been able to get pure vitreous ice in those regions. And so they probably have microcrystalline ice. I could answer that question by taking the grid from the cold stage and free substituting and embedding those and actually measuring the size if there's ice crystal damage. But I haven't done that. Yes?
What kind of depth of freezing you can get in the plunge freezing that you do?
In the plunge freezing method?
Yeah.
The question was, what is the depth of good freezing using a plunge freezing method? I've seen in the literature up to 10 micron thick, but with these cells, which in their thickest portions are 3 microns thick, that's as best that I can do, and that may be due to introducing additional thicknesses from the culture medium and not blotting thin enough. So I would say in my hands less than 5 microns. Yes. Carolyn?
Eileen, we have some copper block and propane jet freezing available here.
With respect to the decoration of surfaces in order to see them well, we've done decoration of carbon replicas with small crystallites of material and found that this introduces a three-dimensional artifact because the contrast is caused by diffraction. And when you change the tilt angle to take the stereo pair, you actually get some of the specks disappearing and others reappearing, and as a result, the tilt action changes the diffraction on the small crystallite. So as you get small and more perfect in your crystalline nature, you're in trouble again.
Do I need to repeat that?
If you want.
The comment was that when you use crystallites, small crystallites to decorate, when they get very small and then they're single crystals essentially, and you tilt, that the contrast of these depends on the orientation of the crystal with respect to the beam, and some of the crystals disappear and some of them reappear. Now that doesn't happen with these amorphous gold. I guess they're amorphous, or anybody know if they're not amorphous? I will assume they were amorphous gold particles. David Agard.
A little bit of that with the small golds in your chronographic data set. Because we go through, we sample a lot of angles, you can actually see them flickering in and out versus tilt, when we do it in practice.
Dr. Agard commented that with the very small gold particles, they see the same effects. How small?
5, even 5.
Even 5,
Even 5.
yeah.